Getting a Doctor of Philosophy degree is considered to be the highest academic achievement, where the PhD degree holder is recognized as an independent researcher, an expert with in depth knowledge of the chosen field of study. But those 5 to 6 years of struggle is the price you pay to get that degree, to get that experience that makes you eligible to do further research and make important contributions in science. And in that period, there's a lot of peer pressure, competition, and a lot of struggle to come up with novel and creative ideas to solve real world problems. All these years, starting from school, then earning college degrees such as MSc, MPhil, and finally enrolling for a PhD, you were trained to accumulate knowledge in the form of theories, concepts, applications, laws, and prepare yourselves for many competitive exams. And then you join for a PhD program just to realize that you have transitioned from a student to becoming a scientist who has to now contribute new knowledge in your area of research. PhD is all about making new discoveries and developing your knowledge of the field. But all of that relies on knowing what others have already discovered and then doing something novel. Here's where we feel blocked. Our creative potential is totally blocked. And if you're someone who's feeling stuck in your work, let's get some inspiration. Hello and welcome to BioBytes for You. And this video is to inspire you to get into that creative workflow. So let's focus on some useful tips from the Nobel Laureates in the field. Let's start with Martin Scharfe, who shared the Nobel Prize in 2008 with two others for the discovery and development of green fluorescent protein, GFP. What did his research experience teach him? His tip number one is ask a lot of questions. Most of us learn biology in school and college in a passive way and accept things as they are. But here's where we need to be like a 5-year-old kid who is curious to know and keeps on asking a lot of basic questions. For example, for a decade after the discovery of DNA, the central dogma in molecular biology was thought to be unidirectional inflow, that is, from DNA to RNA to protein. But somewhere down the line came a scientist named Howard Temin who questioned this flow of information and added to the body of knowledge that information flow could also happen from RNA to DNA. And along the way, he discovered one of the most important enzymes in recombinant DNA technology, and that is the reverse transcriptase. So the cue here is that you need to ask why things are the way they are. Is there any alternate solution or another way to approach the problem? So start with a broad research interest, then keep asking questions and probing deeply into each one of them till you find a specific question interesting that's not yet been answered. Now let's move on to his next tip. He says you should think of different angles you could use to address the problem. Let's take the example of GFP itself. Osamu Shimomura was the first to isolate GFP from the jellyfish. Following his discovery, Martin Scharfe got an idea after attending a seminar that he could insert GFP gene into the ringworm C. elegans and track the movement of GFP inside the cells. Then came Roger Cn, who developed a rainbow of probes based on GFP, which could be used to track multiple pathways inside the cell. So the point here is it's the same GFP discovered in jellyfish, but two very different applications. Number one, use of GFP inside cells as a tracker. Number two, generating different colored probes of GFP through mutagenesis of the GFP sequence. Moving on to his tip 3. He says that you should do experiments in different ways. What are the different ways? Well, this can be done by adding or substituting or combining different concepts and generating new ideas. The best example for this is the Sanger sequencing technique, which employed the concept of substitution. That is, instead of using a DNTP, he used DDNTPs to terminate the chain reaction. So, just by changing, substituting, and including this new component in the reaction, he developed the idea of DNA sequencing. Another example is the reciprocal cross that we study in Mendelian genetics. Reciprocal cross ensures that the results are uniform when a cross is done in both the ways. But what if the results are different for a reciprocal cross? That could potentially give rise to a new knowledge. If you are aware of any such experiment, then please comment and let me know as to how did it add a new concept in biology. And if you don't remember, it's okay. If you want me to make a video on that, please comment and let me know that as well. Now let's move on to the tips by Elizabeth Blackburn. She received the Nobel Prize in Medicine in the year 2009 for her discovery on how chromosomes are protected by telomeres and the action of the enzyme telomerase. Her tip number one is curiosity will develop our creative thinking. Now, how do we develop this curiosity? There are four aspects to this curiosity. First, you need to be resilient when things do not work your way, which means you should quickly recover from your disappointment if you didn't find anything after the experiment and spring back to your research to execute your next idea. Second, you need to be opportunistic. When you see an unexpected observation, which means that you found something interesting, then you should go ahead and pursue that further. Don't discard those findings. Try to be opportunistic about the findings that you've made. Third, be creative. Do this by how you can perform the experiment in different ways to address the problem. Let's say you could do concept A plus concept B, or from the concept AB you subtract concept B, or you combine concept A and B. So, whatever combination works, figure that out. And finally, be persistent. Keep on doing your research experiments with different variables till you find the answer to your research question. Remember, answers won't come right away. You have to work for it, and that's why it's called research, isn't it? Now, let's move on to the tip 2. Her tip number 2 is if you are not failing, you're not pushing boundaries. That's good news for researchers because we often get dejected by failures. But when we understand that failure is the key to success, then we can condition our mind to think like a detective. If this is not working, then it's something else. And then you start exploring another hypothesis, and eventually something will definitely click and it makes sense to you. And then you start further experimentation to develop a working model to best explain that system. You see, failure gives us a chance to be creative and explore all the possibilities that could exist in there. I'm sure you've read about how Edison successfully developed the light bulb after 1,000 failed attempts. It's not about the success that you get, but about the lessons learned from your failure that defines the research spirit of a scientist. Now let's move on to her final tip, which is do research in your own way. You should not be spoon fed. Figure out your own way of doing things, and when things unfold in unexpected directions, that becomes very interesting. Now, this is like a crime series and detective stories. You know, the plot gets interesting, and when things unfold in unexpected directions, that keeps you hooked. Same is the case with our research. You should be in wonderment and be thrilled about your next observation. After all, chance favors the prepared mind. Now, let me introduce you to some new terms here. Have you heard about the word deja vu? It literally means already seen and conveys a feeling of familiarity as if The moment being experienced has already happened. But now, let's reverse that. Suppose you are in a situation that's very familiar. Perhaps you're doing the same kind of experiments and the routine lab work that you have been doing for many days till now, and you suddenly feel as if you're experiencing something completely new. Now, this is called Wuja Day, and it could be a key to becoming a creative, innovative thinker. Let's take the example of Mendel, who analyzed around 10,000 seeds to come up with Mendel's laws, or T.H. Morgan, who was counting tens and thousands of flies to come up with a chromosomal theory of inheritance. What's common? They were doing their routine lab work, and yet they experienced something new. They figured out new patterns by using their own way of thinking. So you should strive to be a lifelong learner. Eventually, you will see a pattern. Till then, keep enjoying the work you do. The next in line is Venki Ramakrishnan, who was awarded the Nobel Prize in 2009 for discovering the precise structure of ribosomes, the molecular machines that make proteins inside the living cells. His tip number one is science is a marathon, not a sprint. Just like you are boosting your general fitness by building stamina in a marathon, in the same way, science is also about building your observations over a period of time, and then you get a big picture, just like fitting the pieces of the jigsaw puzzle. When you practice analytically for years together as a student of science, later, when you become a scientist, your years of practice will make you perform intuitively. That's when your years of experience makes you an expert in the field. You reach a state of mastery when you've absorbed the knowledge so deeply that it becomes a part of you, and then the breakthrough happens. So that's what a PhD life prepares you for. Now let's focus on his tip number two. He says that we should have a work life balance. I'm sure you've all heard the proverb, all work and no play makes Jack a dull boy. And in the same way, your life becomes boring if you don't have a proper work life balance. So keep your hobbies alive. That'll turn on your creative spirit and lead to new ideas. Music is one of the ways that you can turn on your creativity. So do things that unleash your creativity. In fact, Einstein himself has mentioned that his love of music could have played a key role in his cognitive development and in his contributions to science. He says that you should feel your work is important and interesting. When you feel that your work is important and interesting, you will become very creative. Many times, people leave their ideas behind thinking that it's not worth the time and effort. And some years later, someone else publishes the same idea in a high impact journal. And that's when you feel so miserable and regret for not having faith in your own work. So, no matter what your area of research is, be excited with your own work. Make it interesting such that it triggers the dopamine that you need to take your research forward and make breakthrough discoveries. Now let's dive into the tips given by Jennifer Doudna, who along with Emmanuel Charpentier was awarded the Nobel Prize in the year 2020 for their development of the CRISPR-Cas gene editing tool. So here's her tip number one. Good ideas come from everywhere. Even today in research, in some places there's a little bit of bias towards men. But according to her, good ideas come from everywhere, whether it is a male or female. And now we could extend this logic to IQ as well. Many people consider themselves as smart based on their IQ scores and the exams that they have qualified. That sometimes they wouldn't consider the opinion of other people who are not to their level of competence. But the truth is that a brilliant idea can come to anyone at any time, Remember, an IQ score is your data point frozen at one particular time when you took the test. But every day we are evolving and learning and adapting to new things. So, anytime anyone can come up with great ideas. So, the point is if you are doing teamwork, don't dismiss another person's point of view just because you think that person has lower IQ than you, or he or she is not successful and intelligent like you. Her tip number two is that you should focus on basic research. When you answer basic questions, in her own case, when Jennifer Doudna was interested in bacterial immune system, CRISPRs, and was trying to decipher the mechanism of bacterial immunity, that's when serendipity happened in a conference when she met Emmanuel Charpentier, who was working with Cas9. The combination of the microbiologist and this biochemist led to the development of the precision gene editing tool CRISPR-Cas. Her tip number three is that you must solve a lot of puzzles because in her case, it really taught her about the joy of finding things out. To her, it was like having a hypothesis about what the code is and then test it by experimenting on it. That gave her the idea of setting up her own experiments. She says those things were the ones that really drew her to science in the beginning. The next in line is Randy Sheckman, who received the 2013 Nobel Prize in Physiology or Medicine for his role in revealing the machinery that regulates the transport and secretion of proteins in our cells. His tip number one is that you should collaborate your work. When many people's ideas come together, it's going to generate a launch pad for a massive breakthrough to happen. Human Genome Project is one of the greatest achievements in biological sciences, which focused on collaborative work, where several labs work together to obtain the nucleotide sequence of the 23 pairs of chromosomes. Isn't that a great feat in science? His tip number two is to connect two ideas in unexpected combination. Let's look at Paul Berg's idea, the father of rDNA technology. When a lot was known about the bacterial gene transfer by phages, he wondered whether a comparable virus mediated gene transfer system existed for mammalian cells. He came across SV40 virus, which could integrate its DNA into the mammalian host genome. He thought, could it be possible that SV40 could be used to transfer genes from one mammalian cell to another in much the same way that phage transfer genes among bacteria? But the probability of acquiring a specific mammalian gene in a SV40 particle was lower than the probability of a lambda phage particle containing one or more specific E. coli genes. Now, what could he do? He thought of deliberately combining SV40 genome and phage genes in vitro. And this is what gave birth to the field of recombinant DNA technology. Thus, any piece of DNA covalently linked to the SV40 DNA could be integrated into the chromosomes of the mammalian cell along with the SV40 DNA. Learn to connect two ideas. In this case, idea 1 was using SV40 and idea 2 was combining it with a foreign DNA in vitro and then introduce it into the mammalian cells. Now let's move on to his tip number 3. Don't have preconceived notions about your work. There'll be times when things don't work and people around you will discourage you from what you are doing and probably also make fun of the choices and decisions you made in your life. Don't give in to that. This way, you will be cursing yourself for making wrong decisions in life and ruin your research career. The research career that you were most passionate about at one point in time. So be optimistic. In fact, as we've already seen, failure is the key to setting you up for success. Now let's move on to Frances H. Arnold. She was the one who developed directed evolution to create novel enzymes with improved properties and novel functions. She disrupted the conventional rational design approach to building novel enzymes and proteins and was awarded the Nobel Prize along with two others in the year 2018. Her tip number one is built by adding and recombining what is already there. When you recombine the same things in new ways, you get a new solution, new application. Let's take the example of tangrams. These are pieces of wood in different basic shapes that can be combined in different ways to get different shapes altogether, right? Now let's take a scientific example. It's the classic example of zinc finger nucleases. This was achieved by combining the DNA binding domain, which is the zinc fingers, and the cleavage domain of the restriction enzyme, fork 1 endonuclease. Let's move on to her tip 2, co create with nature, which means that you first learn to imitate nature and then innovate. She says that we should become a co creator because nature has explored only a tiny fraction of life's molecules that are possible. With mutagenesis tools in our hand, we can accelerate evolution and create genetic diversity in the lab, and we can explore paths that nature has left unexplored. So learn to imitate nature, and then over time, you will innovate and have the answers to solve real world problems. Let's move on to her tip number 3, be adventurous. Step out of your comfort zone. When you leave your comfort zone and get into the adventure zone, that's when serendipity happens and you come to the brink of breakthrough discoveries. In an adventure, you have a large number of events happening which do not have individual significance but occurring together gives a wonderful outcome. This time, the example is a movie. Have you seen the animation movie Up, which is all about the spirit of adventure of an old man and a wilderness explorer, Boy Scout, and both of them together land on an exotic place? The childhood dream of the old man to be in the Paradise Falls. They both together discover the strange 15-foot bird of paradise. But along the way, he also learns about human psychology. His adventure shows him how his childhood hero had turned out to be an evil, vicious thinker. So it's the adventure that brings about serendipity and novel discoveries and also gives us deeper knowledge. Next in line, we have May Britt Moser, who was awarded the Nobel Prize in 2014 in Physiology and Medicine for her discovery of red cells in the brain that functions like a GPS and plays a role in generating a system of mental coordinates by which animals are able to navigate their environment. Her tip number 1 is great discoveries come from team effort. When all the team members cooperate and work together to achieve a common goal, then great discoveries are made. When each member of the team focuses on different aspects of the same theme, then you get a bird's eye view of the work that your lab is doing. You get the big picture. Think about it as a car assembly line, which is a production process that divides up the labor process, each focusing on one aspect of the car. When different units of production line do their job correctly, what you get at the end is an assembled car ready to be dispatched and to be sent to the showroom for purchase. In a production assembly line, you get more things done in less time, and that moves your work by leaps and bounds. Her tip number 2, trust your own judgment and go for the really long term goals. There'll be times when you have to trust your own judgment and go forward in research. Here's where I want to read out a poem by Robert Frost, The Road Not Taken. So, here are the lines. Two roads diverged in a yellow wood, and sorry I could not travel both. And be one traveler, long I stood, and looked down one as far as I could, to where it bent in the undergrowth. And the poem goes on. Now, moving on to the last stanza. I shall be telling this with a sigh. somewhere ages and ages hence. Two roads diverged in a wood, and I took the one less traveled by, and that has made all the difference. That's precisely the story of many of the scientists who have made interesting contributions in science. So trust your own judgment and just go for it with all your heart. Her tip number three is listen to conversations and build your creativity. There's an article on social media which says that if you have an idea, have a conversation about it with your friend, then get feedback on it, and then get more comments, and then write your final piece and share it with the world. Same is the case with your research. If you have an idea, discuss it with your close friend, then gradually build conversation with lab members who are interested, and then present your initial findings in your lab meetings, and then get more feedback and compile all your findings into a hypothesis that you could experiment with. And when you have enough data, publish your new findings. Now let's move on to the bonus tips. Tip number one is by Sidney Brenner, who was awarded the Nobel Prize in Physiology and Medicine in 2002 for deciphering the genetics of programmed cell death and animal development. Sidney Brenner has mentioned in one of his interviews, daydreaming and taking action are equally important. Now, we know that daydreaming actually takes us to a brainwave frequency alpha, which is involved in creative thinking. So, maximize your creative thinking by meditating and visualizing your goals. And simultaneously, do not forget to take the necessary action to achieve your goals. The last tip of this video is by Paul Morris, who was awarded the 2001 Nobel Prize in Physiology, along with two others, for their discoveries of protein molecules that control the division of cells in the cell cycle. According to him, you must read papers outside your area of research. You will get new way to solve your research problem by reading papers from outside your area. You can now extend that to reading biographies and autobiographies of many scientists. This way, you can expand your knowledge and will be able to connect different sets of ideas. And now, my question to you is what is your favorite tip from this video? Please comment below.